All right, so I'm going to start by telling you a little bit about who I am. So I am from Mexico, and I like grew up in San Diego, but I go to school here in New Orleans. So I am doing a BS in neuroscience and a BA in philosophy, and I've done uh, I've done two different research projects, and I'm working on a third one. And kind of a thing that's like ubiquitous in neuroscience is very much so this MATLAB. Does anybody know what MATLAB is? I don't know. I'm very used to like having interactions. So if anybody is, just say yes. Um, so I've done TBI research, traumatic brain injury, and I've also done some kind of behavioral research. And we use this program, MATLAB, and I've also used something else called Deep Lab Cut. So let me start by telling you what MATLAB is. So MATLAB it's short for Matrix Laboratory, and it's a multi-paradigm programming language, and it is created by MathWorks. So the way that I would put it is MATLAB is very much so kind of like a calculator, and it just kind of remembers the data that you input into it. That's a very basic idea of how it is used. And you can use it to do statistical analysis, but there are a bunch of other things that you can do. For example, if you're using MATLAB for biology, at least that's how I know how to use it. You can use it for genomics, which I haven't done, but I asked a few friends how they would use it. And this is going to be more like an overview instead of actually going into the nitty gritty. And I do have something that I'm going to send to the host so that I can, so you guys could have this sheet, kind of like an introductory sheet to it. So in genomics, you'd use it to kind of just analyze larger data sets after sequencing. And one of the things that actually works really well is there's this thing called BLAST. And BLAST is kind of like a national database where if you have a set of basically like a set of amino acids, and you want to see what this thing is, you actually can. And this database is all run through MATLAB. And it allows you to just process these things very quickly. And if you're able to use this BLAST website, which is anybody can access, you can actually come up with primers for PCR, which is really important in biology. So that is one of the applications, at least for genomics. And one of the other really cool things that you can do is you can visualize potential secondary structures for a protein. So instead of having to do it by hand, which is what they had to do beforehand, you can just get this program to do it. You can also use it for microscopy and biological imaging, which I also haven't done. So I've done more electrophysiology, and I'll go into that. And for that, you can use it for histology, which is basically staining cells and kind of seeing what the penetration is. And that way you can see how much of a protein is in a specific cell. And you can see kind of like how the form affects the function, which is really cool. But the part that I really have worked on is electrophysiology. So I bring this up because electrophysiology basically is seeing the electrical currents in a cell. And I've used it specifically on neuroscience and neurons, or, ganglia, which is basically just a group of neurons. So what you can really do here is you can put an electrode into a neuron, and that would be an intercellular electrode, which is very difficult because neurons are very small. Or you can put an extracellular electrode where you're getting data from multiple neurons. And so, not to go into deep with biology, which it really isn't that much. So cells, for the most part, are negative on the inside and positive on the outside. And you're measuring what the difference in the electrical current is. So why is this data important at all? Well, what this actually tells us is whether some kind of stimuli is actually affecting it. And so one of the projects that I've done is I've been working with sound and whether mice suppress sound. So this was work done by the Schneider Lab at NYU, which I was able to join. And think about it this way: when you walk, do you hear your footsteps? Not really, right? You're kind of stepping and you don't hear them unless you're like in some kind of wooden, or you're stepping on something wooden or something foreign. So your footsteps are making a sound. You're just not hearing them. So what actually happens is your brain filters these out. It suppresses the sound. And this is a really cool finding because we want to know why it's suppressing this. And well, what you can really do is you can put an electrode, an extracellular electrode, into the auditory cortex and see whether these footsteps are making a difference. Or you can put an electrode into the motor cortex and see whether these footsteps are actually making something. So I have a small little presentation to do with that, which I'm going to go ahead and share. Oh, I guess not. It's okay. So, okay. So one of the ways that the Schneider laboratory works is they put mice on a treadmill and they have
Oh, sorry. If you want, you can. I think I changed the
okay,
axis.
I can share
cool.
now.
Sorry about that.
Cool. Okay, so this is actually a nice little graph that'll kind of show you what I'm talking about. So these mice, they are head restrained, and this is all done through MATLAB. So I'm just kind of showing you what the application is. So these mice, they are head restrained, and they actually have an electrode in their head. And what you're trying to, and they are running on a treadmill. And this treadmill, as they're running, it's actually producing a sound that is not their own footsteps. It's actually virtually soundless. The sound is actually a beep or boop or whatever we actually want. And what we found is that these beeps and boops, when the mice are first stepping on day one, they hear them. There's an effect. Then as time progresses, they actually believe that these sounds are their own footsteps. So this self-generated footstep would be this red one. And as you see, they kind of become more desensitized to it. So these reafferent sounds, as we call them, sounds due to locomotion, get suppressed. And what's really cool about this is that all of this data is collected with MATLAB. And MATLAB is really cool because all you have to do is literally just process this data and you get a graph like this one. And that's kind of how you can use it with electrophysiology, at least. One of the other things that you can do is you can use it, and I don't have an example of this one. In fly labs, often you have to do this behavioral test called climbing, where basically flies are in a vial. And you have anywhere from 30 to 60 flies in a vial. And you're trying to see how many of them get to the top. And normally what you would do, or at least back in the day, you would film these flies try to climb. And then you try to count them in a very tedious manner. But using MATLAB, you can actually make a machine learning algorithm that tracks their movement. And I did want to talk about something else apart from MATLAB, which I thought was really cool and I thought would interest you all. Let's see. Okay. So there's this machine learning algorithm called Deep LabCut, which I actually got to use and combined with MATLAB, you can actually do quite a bit. And Deep Lab Cut, what it allows you to do is it allows you to track movement. And it is created by Mackenzie Mathis at Harvard, if any of you are interested with that information. So it's created by the Mathis Lab, and it is an open source program, and it tracks animal movement. And in conjunction with this in MATLAB, what you could actually do is you can graph movement. And you know, if somebody just told me that, I'd really question, why would you care about graphing movement? Well, with behavioral tests, it's very important. So at least in this experiment, let's say I was playing a sound for mice, and I was trying to see their reaction. Normally what would happen is we like put this little dot on the mouse, and then we're tracking this dot. Well, what actually happens with Deep Lab Cut is we can put, well, the dot is a physical dot. With Deep Lab Cut is we can film the actual interaction and then do the processing later. And what this allows us to do is to just do the experiment, and then we actually have numerical values for behavioral data, which allows us to have much more accurate data. And basically, we can actually just, it's just better data, which at the end of the day means better publications or anything like that. And yeah, I just wanted to talk about those two. If you guys have any questions, I'm free to answer them. Does anybody, you can type questions in the chat, can you? Maybe.
Yeah, we can.
Okay, does anybody have any questions? I can answer anything about basically like how I've used MATLAB, which has been mostly electrophysiology, or I can answer any other pressing questions. Okay, so actually, can somebody fill me in on what your project is specifically? Just like a basic overview. Or I think I'm just going to answer like a more general. The thing about MATLAB is that since it is kind of like this calculator that allows you to store data, it's really nice and it's very easy to learn. So it's used all throughout science and engineering. It's actually kind of the preferred method for engineers. So I had a friend who was doing stress tests on, so I have a friend that's a material scientist, and he was doing stress tests. And basically what he was doing is he was pulling some kind of sponge-like thing and trying to see why this thing was so strong and whether poking holes into it would change its strength. And by actually having this data, he's able to actually graph where there was the most stress or anything like that. You can use it in electrophysiology, like I mentioned, or you can use it to just run statistical analysis. So I have a friend that did a statistical analysis of a game theory problem last summer. Okay, so let's see. So I think it just depends on what kind of issue you're trying to combat. So this is actually like a tough question. So I think maybe it would be very difficult to combat any kind of social issue without any kind of statistical relevance. So one of the things that you can do is, since it's basically like a calculator, you can actually run a bunch of data through it and actually analyze whether it's statistically relevant, see whether there is any statistical relevance to any, let's see, see whether there's any statistical relevance to any disparity, which we actually see. Like, for example, one of the things that you see is that the only statistically relevant factor in SAT scores is wealth. So you see that normally students that are wealthier perform better on the SAT. And you can figure out why. You could go run through all of these data sets and you could find any statistical relevance and then see how you could address it. So I think this would be a tool to see where the disparities are. And then afterwards, you'd look for something to do on a more political or social change. At least that's what I would say. Yeah, so MATLAB is used heavily for neuroscience, but we can also use Octave. So Octave is free and you don't use it for MathWorks, and they're very similar, and you don't have to buy a license from MathWorks. The thing is that MATLAB just has many more features and has many more packages that people have already built for it. So it's just much more convenient. And for the most part, institutions pay for MATLAB. You can use Octave instead, though. They are very similar and only have slightly different ways to code, because MATLAB itself is its own language. And like I said, I'll send a PDF to the host so that you can kind of go through the basics of it. It's very simple to learn. I've had friends that have learned it in a night, and it's, like I said, it's a very powerful tool.